Hi, I'm Jennyne Palaroan.
I'm Kimberly Sayam.
I'm Chariz Zambrano.
And I'm Mariz Ramos. Welcome to Crash Course Genetic Technologies.
As we've said in previous episodes, gene technology pertains to a variety of activities about understanding how genes are expressed, utilizing natural genetics variations, altering genes, and transferring genes to new hosts. This episode will take a closer look at DNA profiling.
First, let's begin with the definition of DNA profile. A DNA profile contains a list of numbers indicating repeat units in each copy of 20 marker regions that are located throughout the human genome. Simply put, our chromosomes have markers to which short DNA sequences are repeated multiple times. Now, what exactly is DNA profiling?
It is the process of analyzing DNA variations for the purpose of identification. DNA sequences, which are usually different in different people, are referred to as genetic markers, as I've mentioned a while ago. And since we know exactly where they are on the chromosome, they can be used to construct DNA profiles. In terms of how DNA profiling is applied, this technique is commonly used to identify the origin of a body fluid sample related to crime scenes, reveal family relationships, and even identify disaster victims. As a matter of fact, DNA profiling is commonly utilized in forensic science for unsolved crimes and the identification of human remains. It is considered as a state-of-the-art procedure to identify individuals based on their unique genetic makeup. And although many people share similar eye and hair color and facial features, their DNAs would still not be the same. Hence, DNA profiling is really useful to solve crimes more accurately. DNA profiling is also said to be interchangeable with DNA fingerprinting in terms of both being molecular methods involved in identifying people depending on their genetic makeup, their focus on polymorphic regions, PCR as one of their main techniques, and their usage of biological samples for DNA extraction. Even still, some claim their differences, one of which is that unlike DNA fingerprinting, a molecular genetic method that identifies individuals from unique DNA patterns, DNA profiling is specifically directed towards forensic techniques for criminal investigations and parentage testing. Nonetheless, both still share some similarities in that their main difference lies in their respective methods and uses.
In forensic science today, DNA profiling is produced as follows. Firstly, DNA is extracted from a biological sample.
Now, STR analysis is incredibly sensitive, so it only needs a tiny amount of someone's DNA to produce an accurate result. As a result, the DNA can be extracted from a wider range of biological samples, including blood, saliva, and hair. Next, DNA profiling does not use restriction enzymes to cut the DNA. Instead, it uses the polymerase chain reaction or PCR to produce many copies of specific STR sequences. Once enough copies of the sequence have been produced by PCR, electrophoresis is used to separate the fragments according to size. Then, each fragment passes by a laser which causes the fragments with fluorescent tags to glow with a specific color. The output is displayed as a series of colored peaks like the image that you can see right now on your screens, highlighting the color and length of each STR sequence. The more STR sequences that are tested, the more accurate the test is at identifying someone. Moreover, other STRs used for forensic purposes are called YSTRs, which are derived solely from the male Y chromosome. This is useful for identifying male perpetrator from mixed DNA samples. Remember, only one person in every 10 million will have a particular STR profile. With the world human population estimated at only 7,100 million, it is therefore extremely unlikely you will share the same profile as someone else, unless you are identical twin.
Now, turning to the benefits and risks of using DNA profiling, let's take a look at this table.
First, let's focus on the advantages of this method. It is highly unlikely that two individuals would share the same profile, except in the case of identical twins as mentioned earlier. Therefore, DNA profiling is considered reliable. Moreover, it can be used to ascertain paternity, identify genetic disorders early, and place suspects at a crime scene. However, genetic profiling can be controversial as it is still not a perfect science. Although it may seem reliable due to a technology that has 99.9% accuracy, errors may still occur when that percentage is applied to real-life cases. Privacy issues may also arise if the information is leaked or not kept safe at the lab, since it can complicate an individual's healthcare, insurance, processes, and job prospects. Most people believe that using DNA profiling violates their privacy and civil liberties. It also raises concerns over third-party access since it can be used negatively. For instance, stealing DNA profiles from a database, planting DNA evidence at crime scenes, ethnic targeting, profiling, and discriminating individuals. That is why we have ethics and regulations for.
Due to the concerns entailing DNA profiling, regulations such as
GDPR and the Philippines Data Privacy Act of 2012 are established to protect individuals' information by enhancing their control and rights over their personal data. GDPR is a data protection and privacy rule in EU law that applies to individuals in the European Union and European Economic Area. The export of personal user data outside the European Economic Area and the European Union is also addressed. On the other hand, the Philippines Data Privacy Act of 2012 or Republic Act 10173 is a regulation formed by the National Privacy Commission that lays out a set of rules for both government and private enterprises to protect personal users' information. This establishes a framework for data privacy, accountability, and compliance that will address a wide variety of concerns, including governance, data security, training, third-party affiliations, and breach reporting. Overall, the goal of both privacy laws is to protect an individual's personal information, including the processing of genetic data, biometric data to identify a natural person uniquely, data concerning health, or data concerning a natural person's sex life or sexual orientation. GDPR protects fundamental rights and freedoms of natural persons, specifically the right to protect personal data while enabling personal data's free movement. At the same time, data privacy law provides free flow of information while protecting fundamental human rights. In addition, there are a variety of fines that can be imposed under the data privacy law that is dependent on the nature of the damage or the conduct committed.
To recap, we saw DNA profiling as one of the great discoveries in recent history. This technique determines an individual's DNA characteristics by getting a sample of an individual's DNA and then analyzing and comparing it with another sample to know whether there is a genetic match. Although DNA profiling has great benefits, risks and issues regarding this technique remain. Hence, following ethical laws and regulations is of vital importance.
Next episode, we'll learn about CRISPR gene editing as we begin to explore other forms of genetic technology.
Until then, thanks for watching this episode of Crash Course on Genetic Technologies DNA Profiling.
If you want to help keep all crash courses free for everybody forever, you can join our community on Patreon.